So today we'll be doing with the next part of the plasma membrane. We have completed the structure. We have understood the Singer-Nicolson model. We have got a brief idea about the different biochemical components present within the plasma membrane. And now we move to another part of the plasma membrane that is very vital with respect to the fact that all biological cells are covered with a biological membrane. And the membrane actually controls what molecules can move out, what molecules can move in. So it's the permeability of the membrane that controls the entry and the exit of biomolecules from within the cytoplasm to the exterior and from the exterior to the cytoplasm. So for reference, I have taken quite some material from Albus, Leninger, Cooper. You have heard about these books. This is another book. This is a good one. Cell, Pollard. Then Lorish. Lorish is also a good book, but it's a bit higher. So if you want to study Lorish, you need to study Bruce Albus first. Then this is a good book for biochemistry, Garrett, Harper, and Matthews. Now, let's look into this picture. This picture is specifically the left one is giving us a very schematic representation of the different category of molecules present in nature, different category of biomolecules present in nature. And if you look, we have this hydrophobic molecules. The hydrophobic molecules includes oxygen, carbon dioxide, the gases, nitrogen, and the steroid hormones. Then there are these small uncharged polar molecules. The moment we are saying that they are polar molecules, they are much soluble in water, but they are small, uncharged. So we have water, urea, glycerol, ammonia. Now we are coming to large, uncharged polar molecules. So this is glucose and sucrose. And finally, we are coming to ions. Now the basic concept is that if given enough time, any molecule will diffuse across a plasma membrane down its concentration gradient. So usually the molecules have a tendency to diffuse through the plasma membrane down its concentration gradient, means that this side of the membrane should have a higher concentration of that given biomolecule, and the cytosolic side of the membrane will have a lower concentration of the given molecule. So that is down the concentration gradient. Now, the rate of diffusion, how fast these molecules are diffusing through the membrane varies, and it depends on the size of the molecule. That is evident because smaller the size, the faster it diffuses through the membrane. So the rate of diffusion is dependent on the size of the molecule. That is one thing. And on the relative hydrophobicity. Now, what do we mean by hydrophobicity? How much it is soluble in oil. Remember, the membrane is a lipid bilayer. So the movement of molecules through the membrane is partially, or I wouldn't say mostly, dependent on the size of the molecule. Smaller the size, faster it diffuses, and on how hydrophobic the molecule is, how easily it can diffuse into oil, how easily it can be soluble in oil. So given these two concepts, so we developed two concepts, that is size. Size is an important point when molecules move across the membrane. And another important point just now, as I told, is the hydrophobicity. So considering these 2 points, just a second, let me write this hydrophobicity. So size and hydrophobicity are the 2 main determining points which determines how fast a molecule will travel across the plasma membrane. So the general rule is smaller the molecule and more hydrophobic or non-polar it is, more easily diffuses across a lipid bilayer. So if you look into the first category, you see The small non-polar molecules such as oxygen carbon dioxide can readily dissolve in lipid bilayer and can diffuse rapidly. When we are coming to small uncharged polar molecules such as water or urea, they also diffuse across the bilayer, but much slower as compared to this category. Now, by contrast, when the lipid layers, as I said, are totally impermeable, you can see the lipid bilayer is totally impermeable to charged molecules, no matter how small they are. The impermeability is because of the charge and high degree of hydration, that is a layer of water covering this charged molecules that prevents them from entering the hydrocarbon phase of the bilayer. So in nutshell, one thing we get is that smaller the size of the molecule, faster it can diffuse across the plasma membrane. More hydrophobic the molecule, faster it can diffuse across the plasma membrane. Polar molecules, but smaller in size, can also diffuse across the plasma membrane, but at a slower rate. Ions cannot diffuse across the plasma membrane. So this is what we get about the different molecules. But further, before getting into what are the different transport mechanisms, we need to have a very clear concept about two basic words. One is diffusion and the other is osmosis. So when I'm clarifying basically these two words, diffusion and osmosis, many of you rightly said, osmosis is a movement of solvent molecules from a region of higher concentration to a region of lower concentration, whereas diffusion can be broadly summarized as the movement of solute molecules from a region of higher concentration to a region of lower concentration. Keep that in mind that whenever there is a molecule present in higher concentration at one side and it is gradually moving to a lower concentration on another side, there is always an establishment of a concentration gradient. So there is a gradient. So if I draw a graph graphically, if I show you this picture, so there is a higher concentration of this solute molecule and it is gradually lowering on the other side. So this is known as concentration gradient. So we have a more of one molecule on one side of the membrane and a less of another molecule on the other side of the membrane. So the inherent tendencies of the molecule will be to move from area where it is more concentrated to an area where it is less concentrated. So that equilibrium is established on either side of a membrane. So that is the inherent tendency of a molecule to move. So this is concentration gradient that is established in most cases across membranes. For example, if I tell you here, let us concentrate here. You can see that we are having oxygen, carbon dioxide and nitrogen. They're present on this side more. And we do not have any of these molecules on this side. So what is happening? We have a concentration gradient of oxygen, carbon dioxide and nitrogen in such a manner so that this side they are not present, this side they are present. So they will tend to move in this direction. So this is the movement of molecules along their concentration gradient. If you hear the word that the molecule is moving against its concentration gradient, that means we are having a higher concentration of these molecules on this side. We have a very few molecules of oxygen present on this side, but yet oxygen is tending to move on this side. That is, oxygen is now having a propensity to move from a region where it is less concentrated to a region where it is high concentrated. So this category of movement is known as against the concentration gradient. So when we will be understanding the function of the plasma membrane will be constantly coming across this line. It is moving down the concentration gradient. it is moving against the concentration gradient. So be clear, down means we are having a higher concentration, it is moving towards a lower concentration. And up means, against means, it is moving from a region of lower concentration to a region of higher concentration. So I think so we have clarified the basic concept between diffusion and osmosis, and we have clarified two definite words, down the concentration gradient and against the concentration gradient. Now, if I come to the function of a membrane, you can see that the main function of a membrane is transport. So it plays a big role in transport of molecules from one side to another side. You imagine a cell. A cell is covered by a biological membrane. A cell cannot live in isolation. A cell has to constantly crosstalk with its exterior environment. How can it crosstalk with the exterior environment? It can only crosstalk with its exterior environment if molecules from outside move inside or molecules from inside move outside. So in a way, the membrane is actually facilitating the transport of molecules from the exterior of the cell to the interior of the cell and vice versa. When we are talking about transport of molecules, it can be small molecules or it can be large particles. When I said small molecules, we started with these small molecules. So they can be carbon dioxide, oxygen, nitrogen, and so on. And when we are talking about large particles, it can be, just a second, it is not the ions. When you're talking about the large molecules, I wanted to bring the cursor to the glucose part. And when we are talking about large particles, we can say that it can be glucose, because glucose is one of the essential food that needs to enter every cell, so that you all are aware of the glycolytic, the Krebs cycle pathway, so that carbohydrate metabolism can take place and ATP can be generated. So when we are thinking of a membrane, the first thing is that we understood that it is involved in transport, and it can transport two categories of molecules, the small molecules and the large particles. Let me restrict to the small molecules first. When we are talking about small molecules, there can be two ways in which the molecules can move in and out. One is passive, and you can see it's diffusion. That is, the molecules are simply moving from a region of higher concentration to a region of lower concentration. Keep it in mind that there is a plasma membrane present, and we are talking about molecular movement across the membrane. So let this be the exterior of the cell, and let this be the cytosolic part of the cell. So we are talking about the movement of molecules across the plasma membrane. So small molecules can move via passive diffusion, as in the case of oxygen or carbon dioxide, where the molecules are moving from a higher concentration to a lower concentration, so that's diffusion. But molecules can also move from lower concentration to higher concentration. They can be forced to move from a lower concentration to a higher concentration, and then we are talking about active movement of molecules. The moment we are talking about active movement of molecules, you understand a molecule is moving against its concentration gradient. So it has to be forced to move. So sort of energy is essential to drive this process. So active transport invariably involves ATP. A beautiful example of active transport is the sodium-potassium pump. I'll show this mechanism later on. I'm just giving a description of the different categories of transport that is taking place first. So we are thorough that small molecules can move passively down the concentration gradient or actively against the concentration gradient. Whenever the small molecules are moving against the concentration gradient, they require energy. Now coming to diffusion. As you can see, again, we are having three categories: simple diffusion, facilitated diffusion, and we have a third category, that is the osmosis. When we are talking about simple diffusion, what happens? the molecules are diffusing across the bilayer. No help is required. And when I'm using the word diffusion and it falls under passive transport, see, I mean, using the word diffusion and it is falling under passive transport. So remember, simple diffusion is the movement of molecules from a higher concentration to a lower concentration. No help is required. But again, we are seeing the term facilitated diffusion. For example, we have ions. As I started today, that ions are impermeable to this plasma membrane. They cannot move by themselves. We have a higher concentration of ions on this side. No ions present on this side, but the ions need to move. So somebody should facilitate them. So the ions are moving from a higher concentration to a lower concentration, but they are being held by a carrier molecule. A carrier is helping them to pass from one side of the membrane to the other side of the membrane. So that is facilitated diffusion, where a carrier molecule is being involved, or a sort of transporter. I will not say that, I will not use the word carrier here, rather use the word transporter. A protein which is present in the membrane, which will help in the transport of the ions from the outer side to the inner side. So that is facilitated diffusion. And we discussed about osmosis. Osmosis is the movement of water molecules from a higher concentration to a lower concentration. Now, when we talked about diffusion, I like to just highlight lastly on these 4 factors. So diffusion is dependent on the initial concentration of the gradient. How much more, what is the concentration gradient? The more the concentration gradient, the more faster diffusion can take place through biological membranes. The size of the molecule, this we discussed, smaller the size, faster it can move through the membrane. State of matter, whether it is gas, whether it is in solid state. So that is important. And then temperature. If you remember, when we studied the structure of biological membranes, at higher temperatures, membrane tends to become disordered. So they lose their rigidity. So temperature is an important point, because with temperature, we see certain enhancement of movements of the biomolecules within the membrane. So temperature do play a vital role in movement of particles across the membrane. So just a second. So that was about the small molecules. When we are coming to the large particles, as you can see, we are having 2 basic categories, exocytosis and endocytosis. Exocytosis is when you are having some vesicle, or you are having some cellular debris, and the cell needs to throw out this cellular debris to the exterior. So a large vacuole or a cellular debris is moving from the interior of the cell to the outside, exo. So that is exocytosis. And endocytosis is when you engulf something, when the cell is engulfing something. For example, there are certain cells in our body which are the immune cells of our body. Some of these immune cells are capable of engulfing bacteria. So they are engulfing the bacteria. Then they will process and further disintegrate the bacteria. That is a different story. But for our benefit, we restrict to that fact only, that they are capable of engulfing the bacteria. So that is endocytosis. A very loosely term that is used is that when you engulf, it is if it's a solid material, it's phagocytosis. If you're engulfing something of more of a liquid consistency, then it is pinocytosis. So that is about the larger particles. The larger particles, actually the whole of the plasma transporters and the channels that we will study are restricted to the small molecule transport. For the transport of larger particles, there are usually no transporters involved. The membrane folds inwards after engulfing, or the membrane or vesicles, small balloon-like vesicles are formed inside the cell, which fuses with the membrane, and the materials are thrown out of the cell. So we'll come to this exocytosis and endocytosis at the last part of our transport of plasma membrane. So This is one picture. I just wanted to show you this picture, which shows the permeability coefficient of the various molecules through synthetic lipid bilayer. So this is the high permeability. You can see. Let's forget about the writing. Just let's look at the graph. If you check at the graph, you can see that this area is showing the high permeability. You see, oxygen is a very highly permeable gas. And if you look into the lower panel, you can see the charged ions. They usually do not have any very low permeability. Glucose is somewhat intermediate, but towards the lower side. So there is also a transporter present in the membrane that helps in transport of glucose molecules from one side of the membrane to the other side of the membrane. Now this is another way, but this is where the difference has been shown as to how many channels, how many transporters are involved. So some of we have already discussed now. So when we are talking about cell transport, we studied that there were transport of small molecules and large molecules. Okay. So we'll now restrict ourselves that transport of molecules are happening. There is passive transport. No help is needed. Molecules move across the plasma membrane from a region of higher concentration to a region of lower concentration. So if this is my plasma membrane, let us this be. So molecules are moving across the plasma membrane. This is the cytoplasmic part. This is the exterior. So molecules are moving across the plasma membrane from a region of higher concentration to a region of lower concentration. That is passive transport. The moment we are talking about active transport, some sort of energy is being required. So this is the cytoplasm, this is the exterior. Here also the molecules are traveling inward, but this is a ATP dependent process. So as you can assume, the molecules are usually in most cases moving from a region of lower concentration to a region of high concentration. That's a very big difference between passive transport and active transport. When we move on to passive transport, we have simple diffusion. That is the most easiest of the lot, which includes osmosis. When movement of liquid molecules occur across the biological membrane from a region of high concentration to a low concentration. Simple diffusion can also take place in case of gases. When we're talking of facilitated diffusion, as I said, many molecules can move from a region of high concentration to a region of low concentration, but it becomes difficult for them to travel through the bilayer because the bilayer is hydrophobic in nature. It is basically made up of lipid molecules. So there are these channel proteins and carrier protein present in the membrane, which bind with these molecules and help them to transport across the membrane. So that is facilitated diffusion. Coming to active transport, we have vesicles and pumps. Pumps are specifically used to transport ions from one side of the membrane to other side of the membrane. So you must have heard about the sodium potassium pump. We have a lot of other pumps also. So pumps are transporting charged particles from one side of the membrane to another side, and invariably the pumps requires the function of the utilization of ATP molecules. When we talk about vesicles, vesicles are nothing but small balloon-like bodies that are formed within the cytoplasm. This balloon can be formed by partial budding in of the plasma membrane also. I'm not going to get into details of vesicle formation right now because at the last of the fag end of this topic, I'll be showing you how vesicles are formed. But just now I said that vesicles can be of vesicle formation takes place, and there can be exocytosis and endocytosis. Exocytosis, as I said, is throwing out of cellular material from the cytoplasm to the outside. Endocytosis is engulfing of materials from the outside to the inner side of the cell. So this is about the cellular transport. Now, if we look at the transfer of material and information across the membranes, so we discussed diffusion, that is passive and facilitated. We just now discussed active transport. When there is cross-membrane movement of large molecules, we studied it can be vesicular movement that involves endocytosis and exocytosis. When we are talking about signal transmission, I'll not get into the details of this one right now because you have signaling in your syllabus. So I'll be talking about this signaling part thereon, but there are cell surface receptors present on the membrane, which can bind to small molecules on the exterior end, and this binding induces a conformational change on the cytoplasmic surface of the receptors. So during signal transmission across the membrane, most often this case happens, or it so happens that a signal binds to the receptor on one side. Some small molecules are attached to the cytoplasmic end of the receptor on the cytosolic side. These small molecules are activated. They move away from the receptor and further activate a downstream signaling molecule. So when I was talking about amphitropic proteins, I said this happens. And there are certain receptors, as in case of steroid hormones, these receptors are present in the cytoplasm itself. They're not found in the membrane. So what happens, these hormones, they can move through the lipid bilayer and can bind in with the receptors present in the cytoplasm itself. So the steroid hormones move across the membrane, which is nothing but a form of diffusion. And now coming to intercellular contact and communication. As I said, passive diffusion is a flow of solutes from a higher to a lower concentration. Facilitated diffusion is passive transport of a solute from a higher concentration to a lower concentration, which is mediated by a specific protein transporter. So any molecule, any protein molecule that is facilitating in transport of molecules across the membrane is known as a transporter. Transporter can be again of various categories. We'll discuss that later on. Active transport is the movement of solute across a membrane in the direction of increasing concentration. So that is against the concentration gradient, and thus it requires energy. And in most cases, as I said, a specific transporter has to be involved or pumps. And coming to extracellular, last is the extracellular microvesicle exome secretion. We'll talk about the later on about the movement of larger particles. So once more, recapitulation of what we have done till now. Membrane transport of small molecules, we have passive transport. From passive, it is simple and facilitated. Simple is simple, nothing to be done. For facilitated, we have transporters of various categories, and we have channels passing through the membrane, through the entire length of the membrane. These channels helps in transport of ions from the exterior to the interior. And talking about active, mainly these are again pumps, which utilizes ATP to move molecules against their concentration gradient. Now, let us have a basic concept about these three words: an isotonic solution, a hypertonic solution, and a hypotonic solution. So when we talk about isotonic solution, see the solution that has the same solute concentration as another solution. So let's look into this picture. So we are having 20% solute concentration, 80% water, exterior also 20% solute concentration, 20%, 80% water. So within the cytoplasm, the total concentration of the solute is 20% and the remaining 80% is water. Assume it to be a hypothetical condition. And on the exterior environment also, we are having 20% of solute concentration and the remaining 80% is water. In such a scenario, what happens, since the solute concentration on either side of the membrane, this is the cell, so there is a membrane. So on either side of the membrane, the concentration of the solute is constant. Inside the cell, it is 20%. Outside the cell, it is 20%. So that is equal. We are having equivalent solute concentration inside the cell and on the outer exterior solution. Hence, no movement of So, as you can see, there is no net movement of water molecules as the overall concentration of the solute is same on either side of the membrane. Such a solution is known as isotonic solution. So in isotonic solution, this is important, no net movement of water particles takes place. Most cases you must have seen, must have heard about this fact. The saline is an isotonic solution, not 0.7% of normal saline is an isotonic solution. So this is about isotonic solution. Now we're coming to a hypertonic solution. Look at the picture. We are having, this is the interior of the cell, 20% solute concentration and 80% water. And consider the exterior environment of the cell. We are having 60% water. And the solute concentration is 40%. So this is a more concentrated solution. Okay. So if this is a more concentrated solution, the immediate tendency of the system will be so that the concentration of the solute and the water within the cell should equal to the concentration of the solute and the solvent outside the cell. So as you can see, we are having a higher concentration of water molecules inside the cell. There will be a tendency of the water molecules to move out from the cell so as to increase the water concentration outside the cell, thereby decreasing the total concentration of the solute outside the cell. So these types of solutions are known as hypertonic solution. So you place a cell, when you place a cell in a hypertonic solution, keep it in mind that water molecules will move from within the cell to the exterior of the cell because the cell has been placed in a hypertonic solution. So the cell will tend to cringe. So you can see it causes crenation. And finally, if you place a cell in a hypotonic solution, so you have guessed it by now that a hypotonic solution will have more water concentration as to the interior of the cell. So again, as I said, the tendency of the system will be such that the concentration within the cell and the concentration outside the cell, they tend to be in equilibrium. So as a result of which, to equalize the concentration of the solute and the solvent on either side of the membrane, water molecules will move to the interior of the cell if the cell is placed in a hypotonic solution. And as water starts moving in, you can understand the cell will start swelling. The cell will start to swell. And gradually a time will come, if more water influx is allowed, the cell will burst out. So the cell will lyse. So these are the three standard solutions that are experimentally that can are observed. And that is how a cell behaves. When you put a cell in an isotonic solution, no change. In a hypertonic solution, it starts to cringe, it starts to shrink. And in a hypertonic solution, it starts to swell. So as I started saying, with the various category of transports and the various molecules that are transported, so you have now familiar with simple diffusion and facilitated diffusion. Now look at the picture. So it is a movement of, in simple diffusion, a molecule is moving from outward to inward down concentration gradient. When we are talking about facilitated, again, it is down the concentration gradient, but you can see there is a green color molecule present in the plasma membrane through which our molecule of concern is moving. So this green color channel, or it can be a transporter protein, is helping the molecule to move inside the cell. When we are talking about ion transport, as I was saying, ions are impermeable to plasma membrane. We have these molecules known as ionophores. Ionophores can bind to ions, they pass through the bilayer, they liberate the ion, and then the empty ionophore again comes outside. So this is how the ions move across the bilayer. There's another way by which ions can move across the bilayer is the presence of ion channel. But here As I said, this is again diffusion because the ions move from a region of higher concentration to a region of lower concentration. So it is moving down this electrochemical gradient. See, this is down the concentration gradient. That means higher concentration outside, lower concentration inside. And this is written as down the electrochemical gradient. That means ion is what is ion? It's a charged molecule. So when we have a huge amount of ions on one side, a sort of charge is residing on that side. For example, imagine there are plenty of sodium ions on the outside. That means huge amount of positive charges on the outside as compared to the inside. So the word electrochemical. So there is an electrical gradient also present. Hence the word is written as electrochemical gradient. When you're talking about concentration, a solute particle is not exhibiting any charge. When we're talking about electrochemical gradient, remember that the molecule that is being transporting is bearing a charge. So the charge is now also playing a role in its movement from a region of higher concentration to region of lower concentration. Now we are having, we also studied the active transport. And during active transport, I said that ATP is involved. So this is active transport, we have ATP. So the molecule is moving from a higher concentration to a lower concentration, and you can see ATP is broken down. We have another concept known as secondary active transport. The secondary active transport is that the movement of one molecule is actually facilitating the movement of another molecule. Let me explain it. If the ion is moving down its electrochemical gradient, that is, it is moving from a higher concentrated region to a lower concentrated region, this movement will facilitate the movement of a solute molecule from a lower concentrated region to a higher concentrated region. Iron is in a higher concentration. This side it is lower concentration. The solute, this side is higher concentration. This side is low concentration. So since the ion is moving from a higher to a lower concentration, this movement is helping the movement of the solute from a lower to a higher concentration. Here ATP is not used. So this is secondary active transport. So we'll stop here today.